I'm going to press record. So we are recording now. We are in talk 11 of our 20-part series. So we are on the second half, and so today we'll have the 2, and then next week we'll have 4, and then we'll have 4 right after that. So after today, our next talk will be on next Tuesday at noon, and that will be Matthew doing that talk, unless I got it wrong. I don't think so. I don't have it open though. Otherwise, I'm going to pass this off to Allison and Jade to do this lovely introduction, and we will hear from them. Take it away, Allison.
Go team! So it gives me great pleasure to introduce Larissa. She has been in the chemistry department since the beginning, since her beginning actually, and 4 years ago at Lawrence. And she's originally from Appleton, which is awesome. And she, she's been working in multiple labs close by. Actually, she worked at UW-Madison. In the Homer group, and then she also worked at UW-Milwaukee for a summer, and just as being part of the chemistry department has been fantastic. She's done incredible work with Axel, so the ACS at Lawrence group. She's always been such a delightful soul for students who are struggling with chemistry. It's like, oh, just go talk to Larissa, and just a student who can really connect students who are struggling and also connect with students who are really succeeding. So Larissa really spans the spectrum of an awesome, just fantastic, well-rounded chemistry and biochemistry student, I should say. So anyway, it's been so much fun to have her in PChem as well. So without further ado, the other thing too is this work should have been presented at the ACS national meeting. Hashtag COVID sad. But she's going places, and we're really excited to see where she goes next. So without further ado, Larissa Davis.
Thank you for your introduction, Allison. So my research was on the impacts of copper phosphate nanosheets on the organism Chironomus riparius. And this was done in the Clapper Lab at the School of Freshwater Sciences in UW-Milwaukee in conjunction with the Center for Sustainable Nanotechnology. There we go. So in this study, I explored how the material copper phosphate nanosheets can impact the organism Chironomus riparius, so that we can take a broader look at how this material may overall impact the environment. But before I really get into that, let's talk about why we care about materials like copper phosphate nanosheets in the first place. And really, it stems to the global food demand and the increase in the global food demand that we see today. So before 1900, we can see that the global food demand remained rather steady across hundreds of years. But then once we hit 1900, that global food demand began to rapidly increase. And we can see between 1961 and 2000, the population had increased by 98%. And with that population increase, the food production also had increased by 146%. This increase in food production to keep up with this population increase was only possible due to the invention of the Haber-Bosch process in 1913. So this is a process that allowed us to chemically augment our soils with nitrogen in order to increase our crop yields. So nitrogen is a macronutrient that's needed by all organisms, including plants, that is very important for growth. And the most abundant source of nitrogen is in the atmosphere as inert nitrogen gas. However, this form of nitrogen provides a bottleneck to plant growth. This inert nitrogen gas is not biologically available, and plants are only able to uptake nitrogen in the forms of nitrate and ammonia. Since this gas is so inert, there are very few biological reactions that can convert this abundant source of nitrogen to ammonia. And one of those organisms that are able to do this are nitrogen-fixing bacteria. So while these nitrogen-fixing bacteria are able to produce enough ammonia from this nitrogen gas to support our plant growth before the 1900s, it was not sustainable enough to really support massive population growth. That is until the Haber-Bosch process came about. So this process was an industrialization of this conversion from nitrogen gas to ammonia. And once we're able to form ammonia using this process, we could then add this ammonia to our soils and therefore increase our crop yields. And we can see a direct correlation between the increase in nitrogen augmentation with the Haber-Bosch process to the human population increase in the 1900s. And this can further be correlated to our increase in global food demand that we see in the 1900s. So we can essentially say that the introduction of nitrogen in our soils through the industrial process of the Haber-Bosch process allowed this population boom. While the Haber-Bosch process was able to support this population boom around the 1900s, our population continues to increase today and is expected to reach 9.7 billion worldwide by 2050. With that population increase, the amount of food required is also expected to increase by 71%. And the Haber-Bosch process is limited and won't be able to keep up with this increase in population. So new strategies are needed to keep up with this population growth. So current research is looking at how to expand beyond macronutrients like nitrogen to micronutrients. The micronutrients are nutrients that are needed in lesser amounts than our macronutrients, but they're still very important for plant health, growth, and development. So some of these nutrients include zinc, chloride, magnesium, iron, cobalt, and particularly in the frame of this research, copper. So copper plays a very important role in photosynthesis, which is needed for getting plant energy and carbon for plant growth and disease resistance, both of which can be used to increase our crop yields. So there have been studies looking at copper as a micronutrient, so applying copper in our soils. So this leaf we see here is copper deficient, and we can see it's quite unhealthy. But it was found that by chemically augmenting our soil with a bulk fertilizer, copper sulfate, our plant health increased dramatically. And we can see in this study here that introducing our copper sulfate as a foliar spray decreased our disease by 86% compared to a control, and in conjunction increased the crop yield by 7 times. While our copper is an essential micronutrient that can help increase our crop yields, we do have to remember that too much can be toxic, and there's a fine line between copper deficiency and copper toxicity. With this in mind, we also have to remember that any copper we add into our environment could potentially end up in our waterways and other ecological systems we don't want our copper to be. In 2017, in a comparative study, it was found that 95% of the copper we apply onto our crops via agriculture doesn't end up in the plants where we need the copper to be beneficial to our plant health, but rather down in the soil and in our aquatic systems. Once this copper is in our aquatic systems, it can come into contact with other organisms like fish and midges, which are particularly susceptible to copper contamination. One way we can try to reduce copper contamination and accumulation is through the use of nanomaterials as fertilizers. So a nanomaterial is a material that has at least one dimension that measures less than 100 nanometers, compared to a bulk material, which has dimensions that measure greater than 100 nanometers. Due to their small size, nanomaterials have a much higher surface area to volume ratio, which we can see on the left here, of 0.06 in this case, compared to bulk materials, which we see on the right, which has a surface area to volume ratio of 0.006. This means that nanomaterials have a higher percentage of their molecules on the surface of the material versus in the center compared to our bulk materials. And this surface area to volume ratio, as well as their small size, impart quite a few unique properties that nanomaterials have compared to their bulk material counterparts. So some of these unique properties include differences in color, melting point and boiling point, hardness, as well as solvent interactions. And these unique properties offer several advantages for their use in nutrient delivery. So one of the advantages includes higher translocation, so easier transport up into the plant. And we can see that in this picture here. So these blue dots represent cadmium bulk material. And we can see that most of the cadmium is remaining in the soil and only a small amount is getting up into the leaves of the plant. In comparison, we have cadmium quantum dots represented by these red stars, and less of these quantum dots are remaining in the soil and more is getting up in the plant where they can provide their benefits to plant health. So essentially, less nanofertilizer is needed to have the same effect on plants than our bulk counterparts. Nanomaterials also have a more rapid uptake via the stomata, which are holes in the plant leaves, which again enhance nutrient delivery through a foliar spray. And nanomaterials also often have a more controlled release of ions than our bulk counterparts. The Hamers, or rather copper nanofertilizers, have been further studied through their comparison on disease progression in plants. So we can see here that our nanoparticle copper oxide compared to our bulk copper oxide has a greater reduction in disease, which shows that our copper fertilizers are more efficient and are better at reducing disease than our bulk counterparts. It's also generally found that nanocopper is less toxic than our bulk copper salts. So not only are our copper nanofertilizers more efficient, but they're also less toxic than our bulk counterparts. The Hamers lab at UW-Madison is currently looking further at different nanofertilizers, particularly copper nanofertilizers. And in their study, they compared copper phosphate nanosheets, the topic of this talk today, to commercially available copper oxide nanoparticles and bulk copper sulfate fertilizer. In this study, they found that the copper phosphate nanosheets were more efficient at reducing fungal disease in watermelon than both our copper oxide nanoparticles as well as our bulk copper sulfate, showing that this material, copper phosphate nanosheets, is quite agriculturally promising and could be used to help increase our crop yields. So why might copper phosphate be a better fertilizer than our copper oxide? So here we see a comparison between copper oxide nanoparticles and our copper phosphate nanosheets. And you can see how this copper phosphate has a sheet-like morphology that's quite different than these particles seen in our copper oxide. Though the mechanism is unknown, it is thought that the sheet-like morphology plays a role in enhancing copper delivery with our copper phosphate nanosheets. The copper phosphate nanosheets are also smaller typically than our copper oxide nanoparticles, with an average thickness of 13 nanometers. And finally, our copper phosphate nanosheets have a more rapid initial release of ions while in a solution that replicates plant sap than our copper oxide. And we can see that in our figure here, looking at the concentration increase over time of copper ions. Compared to our copper oxide nanoparticles seen in the red, we can see that our copper phosphate nanosheets has a more rapid increase in our copper concentration within the solution than our copper oxide. And all of these properties are thought to enhance the copper delivery of copper phosphate nanosheets over copper oxide. While our copper phosphate nanosheets are quite agriculturally promising and they're more efficient at delivering copper than our other materials, we do still have to remember that this material could end up in the environment if we were to use it in agriculture. And there's very little information on the environmental impact copper phosphate nanosheets may have. And this is where my research comes in. So there are many different ways to study an impact a material may have on the environment. But how I began was by focusing on the impact on a single organism at extreme concentrations to determine if further studies are necessary on this material. So I narrowed my focus from the impact of copper phosphate nanosheets on the environment as a whole to just on freshwater systems. I narrowed my focus even more to just the benthic zone of our freshwater system, which is the zone right above the sedimental layer. And I narrowed my research even further to just a single specific freshwater midge species, the Chironomus riparius. And as mentioned, I use a very broad concentration range from 0.1 milligrams per liter to 100 milligrams per liter. And to put that into perspective, the lethal concentration for 50% of chironomids for our copper ions, which is a measure of the acute toxicity of copper, is only 1.987 milligrams per liter, which falls in the low range of our concentration range. So essentially, we're trying to see if there's any toxicity we can see of the copper phosphate within this range. And if there isn't, this material is not really something we need to worry about in general in terms of our chironomids. So again, the goal is really to determine if further studies are necessary. To talk further about the specific organism we chose, the reason why we picked Chironomus riparius is because it's a keystone freshwater species, and it lies in the bottom of the food chain. So any disruption with this freshwater midge could have a greater impact on the system as a whole, including in fish as well as waterfowl. This video is showing a larval form of the freshwater midge that I used in my study, but you may better recognize this as this adult form, which you may often see around lakes and rivers, including Lake Winnebago, Lake Michigan, as well as our wonderful Fox River that we were just talking about. So these midges will lay their eggs in the water, and when these eggs hatch, the larvae will burrow down in the sediment, where they grow and develop for a few weeks before emerging again as pupa and becoming adults. Because these larvae burrow down in the sediment, they are a great established model for sedimental exposure of chemicals and has plenty of established culture and exposure protocols, the American Society for Testing of Materials, as well as the US EPA, that we were able to easily adapt to this study. It was also particularly important that we chose a benthic model for the early studies on copper phosphate nanosheets, because nanomaterials like copper phosphate nanosheets have shown evidence of aggregating within solution, and once they aggregate, they can settle down to the bottom, and we can see that here. There's a thin layer of blue right above our sand layer, and that thin layer is settled out copper phosphate nanosheets. So we can see that if there's any organism in the sand down over here, they're going to have direct contact with that copper phosphate nanosheets. Another reason why we chose this organism is because we were able to conduct gene expression studies to try to identify exactly how this material, copper phosphate nanosheets, may be causing harm to our chironomids, if at all. And the only reason why this is possible is because the genome is fully sequenced. So because the genome is fully sequenced, we're able to pinpoint exact genes of interest that we're able to look at to determine how our copper phosphate nanosheets could be having an effect. So for example, we're able to look at genes involved in hemoglobin, which I'll talk about a little later in this talk, including porphobilinogen synthase, PBGS, which is a gene involved in heme synthesis, catalase, which is an oxidative stress response gene that requires heme, as well as genes that are looking at DNA damage and repair. So these include structural maintenance of chromosomes protein 6 and endonuclease 3, which are both DNA repair genes. And again, the only reason why we're able to look at these genes specifically is because the genome is fully sequenced. So now that I've talked a little bit more in detail about the material and the organism, I can get more into detail about my particular exposure. So my exposure was based on protocols, as mentioned, from the American Society of Testing Materials, as well as the EPA, and then also from previous nanomaterial exposure studies done earlier. So I exposed 5-day-old chironomids to 6 different treatments for 7 days. The first treatment was the negative control, which contains only moderately hard reconstituted water, which is just a form of lab simulated hard water. I also have an ion control that is using 100 milligrams per liter copper sulfate, which, if you remember, is our bulk form of our fertilizer. And the reason why we have this ion control is so that we can compare any effects seen in this ion control to effects seen in our copper phosphate nanosheet treatments. And by this comparison, we're able to see if there are any particular effects we see in the copper phosphate nanosheets that aren't seen in our copper sulfate. And any of those effects we can say are nanospecific. And then we could also compare the toxicities between these two and see if one is possibly better than the other on our chironomids. From there, I exposed our chironomids to 0.1 milligram per liter, 1 milligram per liter, 10 milligram per liter, and 100 milligrams per liter of copper phosphate nanosheets. And again, as mentioned earlier, this was a very broad concentration range using extreme concentrations. So to put that into perspective, here's a range from 0 to 10 milligrams per liter. Our concentration range goes from 0.1 milligrams per liter up to 10 milligrams per liter, and even further off the scale up to 100 milligrams per liter. As mentioned earlier, the LC50 of our chironomids when exposed to copper is only 1.987 milligrams per liter. The limit of copper allowed in US drinking water is 1.3 milligrams per liter, which are both down in the lower range of our concentration. And then the EPA criteria maximum concentration, which is the maximum amount of copper that is allowed in a freshwater system before the issue needs to be addressed, is only 0.002 milligrams per liter, which isn't even within our concentration range. So again, by using extreme concentrations, we're able to tell whether or not copper phosphate nanosheets is a material we really need to worry about. If there's no toxicity seen within this range, we can continue on with further studies on different organisms rather than chironomids. So after 7 days, we collected the chironomids, and the first noticeable impact was on mortality. So in the control 0.1 milligram per liter and 1 milligram per liter treatments, 100% of our worms were recovered alive after 7 days. But once we move up to our 10 milligram per liter treatment, only 62% of our worms were recovered alive after 7 days. Moving even further up in concentration, our 100 milligram per liter copper phosphate treatment showed only 1% of worms recovered alive, and our ion control had 0% worms recovered alive. So this does show that our copper phosphate nanosheets can be fatal to our chironomid larvae. Unfortunately, since these two treatments, our 100 milligram per liter and ion control, had such a low survival rate, we were unable to do any further studies on these chironomids. And what that means for the ion control, we are no longer able to compare any effects we've seen between our bulk copper sulfate and our copper phosphate nanosheets. So we weren't ultimately able to say if any of the effects we saw from the copper phosphate nanosheets were nanospecific, or if they're more or less toxic than our ion control. While we weren't able to do further studies on our 100 milligram per liter and ion control treatments, we were able to go ahead with more studies on our other treatments. So after we've collected them, we flash froze our larvae and imaged them under a microscope. And from these images, we were able to measure their body length and compare the different treatments. So after 7 days, the 0.1 and 1 milligram per liter copper phosphate nanosheets showed no effect on our chironomid larvae. But in comparison, our 10 milligram per liter treatment caused at least a 39% reduction in length compared to our other treatments after 7 days. So this shows that our copper phosphate nanosheets does have a great impact on our chironomid larvae growth. This effect on size is also quite visually apparent when we look at the images of our control chironomids compared to our 10 milligram per liter treated chironomids. We can see how this control chironomid is much, much longer than our little chironomid that was exposed to our copper phosphate nanosheets. We can also notice in this image that this control chironomid also has a much darker red color than this chironomid exposed to 10 milligrams per liter of copper phosphate nanosheets. And we can see how this one is even almost transparent in some areas. This red color that we see in our chironomid is due to a protein called hemoglobin. This protein is also found in human blood and is the same protein that gives human blood its red color. This protein is very important for oxygen storage and transport and provides the oxygen for a variety of processes important to growth and development, including respiration as well as metabolic processes. This is particularly important for our chironomid larvae since they live in low oxygen environments like the benthic layer of a freshwater organism. So they really need this protein to help retain as much oxygen as they need for their growth and development. The difference in color between the control and the 10 milligram per liter exposed chironomids seen in the last picture brought up new questions about the effects of copper phosphate nanosheets on the hemoglobin concentration of our chironomids. And we're able to explore this difference quantitatively using hemoglobin absorbance. Hemoglobin absorbs green light in the visible light spectrum from 495 nanometers to 570 nanometers. So really what this means is since our hemoglobin absorbs green light, it reflects red light. Since anything that absorbs light will appear the opposite color or its complementary color. So again, because it absorbs green, it looks its complementary color red. We can manipulate the absorbance of this green light by isolating just the green channel of our chironomid images and by using something called Beer's law. So this Beer's law allows us to calculate the concentration of our hemoglobin using intensity measurements. So we can rearrange our equation to look like this. In this equation, we have our background pixel intensity, which is the intensity of the pixels surrounding the organism in the image. We have the organism pixel intensity, which is the pixels that make up the chironomid in the image. We have the path length, which is the width of the organism. And then we have the molar extinction coefficient of hemoglobin. And with this calculation, we are able to quantify the concentration of hemoglobin present in our samples. And from there, we can compare the hemoglobin concentration across our treatments after 7 days. So after 7 days, we can see that the 10 milligram per liter copper phosphate nanosheet treatment reduced hemoglobin concentration in comparison to our other treatments, which is what we were able to see in our picture and backs up the qualitative observations. We can further look at how copper phosphate may be impacting hemoglobin through gene expression studies on genes related to heme. So what you see here, some of you may recognize as a central dogma of molecular biology, and it shows how all of the proteins in an organism can be made from DNA. So when a protein is made, this double-stranded DNA, which holds all of the information needed to make every single protein, will be transcribed into a single-stranded RNA, which contains just the coding region for the very specific protein in question. This RNA can then be translated into the protein. This process is heavily influenced by environmental factors, as well as the types and amounts of proteins needed at a given time. So say there's a certain environmental factor that causes this expression of DNA to be increased to RNA. We can say that that process is upregulated compared to a control. If instead the environmental factor causes the decrease of this expression of DNA into RNA, that process is downregulated. And this upregulation and downregulation can be quantified using a method called qPCR, or quantitative polymerase chain reaction. PCR is a method that copies and exponentially doubles the amount of a single specific gene of interest that you want to look at within a sample of genes. And this uses very special protocols that are a little bit more detailed than I'm going to go into today. But I will talk about how PCR works. So say you're interested in a very specific gene of interest within a sample that corresponds to a particular protein, and you have one gene of interest. We can quantify the relative amounts of that particular gene in our sample by counting how many reaction cycles it takes to reach a very specific threshold that we arbitrarily set. So say our threshold is 8 strands of DNA. So after one cycle of our PCR, our specific gene of interest has been copied into 2 strands. After 2 cycles, we now have 4 strands of our gene of interest. And after 3 cycles, we finally reach our 8 strands of gene of interest, or our threshold. Now say, instead of just 1 copy of the gene of interest in our original sample, we have 2 copies of our gene of interest. But we still want to reach our threshold value of 8. So after 1 cycle, we already have 4 strands of DNA. And then after only 2 cycles, we've already reached our threshold of 8. So we can see that the more initial gene of interest we have in our original sample, the fewer cycles are needed to reach our threshold value. And this can be compared across treatments. So say again, we have this threshold value right in the middle that we arbitrarily set. And this green line right in the middle is our control sample. For these samples over here, it took fewer cycles to reach our threshold, which means we started out with more copies of our original gene of interest in our original sample, and these samples were upregulated in comparison to our control. All of these samples over here took more cycles to reach our threshold, and therefore had fewer copies of our specific gene of interest in the original sample, and were therefore downregulated. So this method we were able to use for particular genes of interest related to heme in our particular study. So using this method, we isolated all of the RNA in a sample of flash frozen larvae. So remember, this is all of the proteins that are actively being made at the time of freezing. And then because our qPCR requires DNA, we then copy this RNA into a double-stranded complementary DNA, and then we're able to perform our qPCR. And in this particular study, our genes of interest were PBGS, as mentioned earlier, which is a gene involved in heme synthesis, and CAT, which is an oxidative stress gene that requires heme. So once we've isolated and quantified these genes, we're able to compare them across treatments. So we can see that after 7 days, our 10 milligram per liter treatment of copper phosphate nanosheets causes the downregulation of both PBGS and catalase, and it has less relative expression than our other treatments. In terms of PBGS, which is our heme synthesis gene, this indicates that our copper phosphate nanosheets may be having an effect, particularly on the heme synthesis pathway, which then corresponds to the reduction in hemoglobin we saw earlier, and possibly the reduction in growth. For the catalase gene, this downregulation in the 10 milligram per liter copper phosphate nanosheet treatment suggests that these chironomids are no longer producing the needed proteins they need to combat oxidative stress, leaving these chironomids more susceptible to this oxidative stress, which may also be another harmful pathway that our copper phosphate nanosheets may be playing on our chironomids. We can also look at specific genes of interest related to DNA repair. So again, we've isolated our RNA, turned the RNA into DNA, and then performed our qPCR. But this time, our genes of interest was SMC6 and ENDO3, which were both genes involved in DNA repair. We would expect that if our material is causing DNA damage, that both of these genes would be upregulated to help mitigate that damage and help repair it. And in fact, after 7 days, this is exactly what we see in our 10 milligram per liter copper phosphate nanosheet treatment. Both our SMC6 and our ENDO3 genes were upregulated in comparison to our other treatments. So this suggests that our 10 milligram per liter copper phosphate nanosheet treatment is causing more DNA damage than our other treatments that requires more DNA repair proteins to be made to help combat that damage. So all in all, some of the possible outcomes of copper phosphate nanosheet exposure that we saw include mortality, stunted growth, reduced heme levels, changes in gene expression, including heme synthesis and oxidative stress, as well as DNA damage. And I wanted to include this picture again because I think it's a very good visual representation of what our copper phosphate nanosheets can do to these chironomids. So from these results, we can say that our copper phosphate nanosheet exposure may be harmful to freshwater systems through this particular organism, Chironomus riparius. However, again, we also have to keep in mind that this was a very early study that used extreme concentrations. And this was, again, really to see if more research was needed on our copper phosphate nanosheets. And so we can find that the answer is yes, more research is needed. So some of the further research that can be explored on copper phosphate nanosheets is looking how our copper phosphate nanosheets react to true environmental conditions. So that both the morphology as well as the chemical properties of this material could change depending on a wide variety of factors, including pH, temperature, weather, soil composition. And all of these conditions can also affect how this material is taken up by both the freshwater system itself as well as organisms. Other studies can also look at comparisons of copper phosphate nanosheets to other copper applications. In our case, since our ion control caused 100% mortality, we weren't able to compare our copper phosphate nanosheet effects to effects seen in our ion control. So we weren't able to say if any of the effects we saw were nanospecific or if our copper phosphate nanosheets were more or less toxic than each other. So again, we can't discount the impacts we saw on our chironomids from our copper phosphate nanosheets. We cannot also definitively say whether or not this material should or should not be used in agriculture as of yet. And really, more research is needed on this material, both on chironomids as well as on other organisms. I would like to extend a big thank you to Nicholas Niemuth and Becky Curtis, who are my grad student mentors over the summer, in the Clapper lab where I did this research, Jaya Bragada and the Hamers lab for providing the copper phosphate nanosheets, as well as the synthesis of the copper phosphate nanosheets. Dr. Rebecca Clapper and the Clapper lab, Allison Fletchman and Kim Dixon for being wonderful advisors my 4 years at Lawrence, Deanna Donaghue for providing very valuable feedback, Chem 680, and of course to all of you listening. I am open to questions.
Yay!
Having trouble getting unmuted. Thank you so much, Larissa. I will open the floor for questions, as Larissa has already done. I guess I should say you can just speak up. You can raise your hand as Stefan Debert has done, Stefan, or you can, whenever you get heard.
Trying to follow cool Zoom protocol here. So it looks like one of your big questions here is due to morphology, right? So can you tell us a little bit about more about copper phosphate when it's not in these nano sheets, and really, are we looking at just how do we know anything about how fast or slow the dissolution of these compounds are?
I do not myself. I can say that, of course, our copper phosphate bulk material would be larger than our copper phosphate nanosheets, and they would have other properties based on that. So they'd have other dissolution properties, but I have not seen research that has particularly looked at that in comparison to our copper phosphate nanosheets.
Okay. So, and how, you said that you can, when you do the experiment, you can see the blue nanoparticles actually settle out of solution. But I assume at some point those sheets do dissolve, right? So can you give us a sense of how quickly that happens, just even in the abstract?
Yep, we did start on some dynamic light scattering studies on the concentration of the copper within our solution. So we did find that our 0.1 and 1 milligram per liter did mainly dissolve within our solution, at least from what we were able to see with our very basic early studies using DLS. And it really started settling out more in terms of the 10 milligram per liter and the 100 milligram per liter concentrations. But one of the things we also ran into troubles with in terms of this research was that we were finding that our copper also tended to form other like salt compositions within our moderately hard constituted water. So there's more research that was going to happen after I left on how that might be mitigated and what other ways this could really be measured.
Sure, thanks.
Other questions?
I just want to step in. I remembered from some earlier work that copper phosphate is pretty insoluble in water.
Yeah, I think you're right. But you know, I think that the fact that they're seeing the hard water differences tells us that it's at least coming off a little bit. And when we're talking about these low concentrations, then you know, I think even a little bit of dissolution is going to be important to know about. Yeah, thanks.
I have a question. Not to you, Mom, sorry. If you were going to choose another model species to do this same study on, what would you choose?
So I think I would choose a more pelagic organism to kind of see how the effects of our copper phosphate nanosheets compare between a benthic organism and a more pelagic organism like a fish per se. So you can see how these nanoparticles, how they settle out, could really have a differential effect between different species.
Do you think 7 days is the appropriate amount for this kind of research, or do you expect anything change for a longer period of time?
I think in terms of what we did, I think it was effective in seeing the effects copper phosphate nanosheets may have on our chironomus short term. We were able to see very drastic effects from their 10 milligram per liter copper phosphate treatments. However, further studies can be done that take longer periods of time, in which you can also try to see whether the development of our chironomids is impacted as well. So our chironomids only take really about a month or so to develop. So you can also try to see if our copper phosphate nanosheet exposure also stunts that development as well and keeps them from emerging as adults. So other studies can be done as well. The 7 days was just advantageous to us since it was a fairly short study and I had a limited amount of time.
Graham.
Ryan has a question.
Hi. So is the effect you saw on heme upregulation real, do you think? Like, is a little bit of extra copper beneficial for these organisms? At least if you think that having more heme is good.
I think a little bit of copper is, as we've seen in most organisms, it is essential. So a little bit is necessary. But if you look back, let me try to scroll through to get to that figure. Animations. So we're looking at the PBGS, which was the gene involved in heme synthesis. It does seem, in comparison to our control, we can see that there is some error that prevents us from really definitively saying, but in comparison to our 0.1 and 1, it does look like that our copper phosphate nanosheets is causing a decrease in relative expression compared to our other treatments. And I guess we could also possibly say that the amount of copper in our 0.1 and 1 is upregulating our heme synthesis, which may be beneficial, but I think this would really require a deeper look into these impacts, possibly looking at other genes as well.
So as a follow-up, did your standard hard water, medium hardness reconstituted water, which is a great term, I love that. Does that include copper in it then? Or is this just an artifact of the fact that they were lacking copper, but you added a little bit in?
I don't believe our moderately hard reconstituted water has copper. So that may be a good point where a little bit of copper may have helped them as well.
Thanks.
Other questions? Matthew, gosh.
Okay. My question is there a particular aspect of this research that was particularly interesting for you? Or not? I don't know.
In terms of my biochemistry degree, I thought this was really a cool experience working with live organisms since I hadn't had a chance to do that yet. And so I thought it was really interesting to see just how drastic of an effect a material like this could have on our chironomids, not only within the gene expression studies, but also visually. Like, it was very apparent immediately that this had some effect on our chironomids. And I thought that was kind of interesting.
I just want to throw a comment out that I love that the path length for the Beer's Law was the worm. That's just the coolest thing ever.
Well, my document brought up this protocol, and I was like, I had just never thought that we could even do something like this using something like Beer's Law. Pretty crazy.
Well, if there are no other questions, I think I would like us all to thank Larissa again for such an excellent talk. I would also like to invite you all to the talk immediately following this talk, which is Victor's talk. I have sent the link to that Zoom session out if that is easier for you. And if you want to hang and say congratulations to Larissa, we'll probably be here for a couple of minutes, but then I will need to transition over to the other talk. So thank you, Larissa. Thank you all for coming. Hopefully see you in a second.
Yep. Hopping off now, hopping back on. Good work, Al.
Yay!
I did have one other question, actually.
Yeah.
Do you, like, how is the image of, like, the two images, like, of the nanoparticles one and, like, the not nanoparticles, like, the bigger, you know what I'm talking about? Do you know how that was taken?
The image itself?
Yeah. Do you know how it was taken? I'm just curious.
Let me take a quick look.
I assume it could be like.
It was some kind of scanning microscope, I think.
Yeah, that's what would be my guess, but I wasn't sure if you knew for sure.
I can find out real fast, actually. It's It says a micrographs and PXRD patterns of our copper phosphate nanosheets. So it looks like it was a micrograph.
Okay, cool. Thanks.
I also had another question I wanted to ask you, which is more interdisciplinary. But I'm just like curious what your thought on this is, because so I feel like we often talk about food security with technical solutions, but there are also other things to think about in terms of like how we regulate the way that farmers can grow things. And I was just like curious if you had picked up any other perspectives on how to address food security other than like nutrients that you were interested in.
That's a little complicated because even if you look back at the Haber-Bosch process, that is a really kind of bad process for the environment as well. That uses a lot of energy. So just the idea of trying to harness these micronutrients that are fairly limited for use in increasing crop yields, I think is a very tricky subject. And as well as even just the idea of introducing these materials into the environment. But I feel like also something has to be done in terms of food insecurity. And this may be just one of the ways we have to go about it. We try to pick the least harmful pathway.
Interesting. Yeah, I was just curious. Good job. It was really awesome. Good job, Larissa.
I enjoyed presenting very well.
So I think I am going to say excellent job, Larissa. I am going to hurry us a bit because I do have to get Victor's open as well. So I'm going to leave. You guys can stay, but as soon as I open Victor's, it is going to shut down on you.
All right, I'm going to go too. Great job, Larissa.
Bye.